plant hormones. They are also called as phytohormones. They are internal factors that promote or modify growth or inhibit growth. Growth promoters are auxin, gibberellins, and cytokinins. Growth inhibitors are ethylene, which is a gaseous hormone, and abscisic acid. They are organic substances produced naturally in higher plants. Hormones are transported through phloem parenchyma. Auxins, synthesized in growing tip of plants. The natural auxin is indole 3-acetic acid, IAA. The precursor of IAA in plants is tryptophan. Primary function is cell elongation. Synthetic auxins are naphthalene acetic acid, 2,4-dichlorophenoxyacetic acid. Physiological effect and applications. Auxin causes cell enlargement. Then apical dominance. Inhibition of the growth of lateral buds is due to auxin. Stimulation of stem and root growth. Induction of multiplication of cells in tissue culture to produce callus. It stimulates formation of lateral and adventitious roots. 2,4-dichlorophenoxyacetic acid kills dicot weeds. Production of seedless fruits like orange, lemon, grapes, banana by induced parthenocarpy. Stimulation of cell division in cambium and also cause early differentiation of xylem and phloem. Auxins promote rooting at low concentration and shoot formation at higher concentration. It causes early rooting in cutting. Naphthalene acetic acid and 2,4-dichlorophenoxyacetic acid as foliar spray induces flowering in lychee and pineapple. It prevents premature fruit falls in apple, pear, and orange by preventing formation of abscission layer. Auxin breaks dormancy in seed and promotes quick germination. Gibberellins. It is synthesized in root tip and developing seeds. It shows non-polar transport through vascular tissue. It was first isolated from fungus. Rice plants infected with gibberellin shows extensive stem elongation called Bacchini disease. Precursor is mevalonic acid. Gibberellic acid 3 is most common and biological active form. Physiological effect and application. Breaking of bud and seed dormancy. Stimulation of seed germination in cereals like barley and wheat by synthesizing hydrolyzing enzymes. Stem elongation due to increase in length of internodes. Promotes bolting and flowering in plants with rosette habit. Example, beet, cabbage. It causes parthenocarpy in tomato, apple, and pear. Stimulation of flowering in long day plants used to increase the fruit size and bunch length of grapes. Increase in the height of genetically dwarf plant like maize. Overcome the requirement of vernalization. Inhibit growth of root. Delays senescence and prevents abscission. It causes production of male flowers on female plant. Cytokinin. Cytokinin from coconut milk is kinetin. Cytokinin from maize is zetin. Chemical nature 6. Peripheral aminopurine. Physiological effects and applications: promotes cell division and cell enlargement. A low cytokinin to auxin ratio induces rooting, while high ratio causes shooting. It delays the senescence, that is aging, breaks dormancy and promotes seed germination, reverses apical dominance and promotes the growth of lateral buds, induces RNA synthesis and formation of interfascicular cambium. Ethylene, the only gaseous plant hormone. Source of ethylene: ethaphon, synthesized in roots, shoot apical meristem, ripening fruits. Physiological effect and application: promotes ripening of fruit like banana, apples and mangoes. Stimulate initiation of lateral roots in plant, breaks the dormancy of bud and seed, accelerates the abscission in leaves, flower and fruits by forming abscission layer, causes apical dominance and inhibit the growth of lateral buds, enhances the process of senescence of plant, inhibit flowering in most of the plants except pineapple, causes epinasty, that is drooping of leaves and flower, increases activity of chlorophyllous enzyme, causing degreening effect in banana and citrus fruits. Abscisic acid, it is a natural growth inhibiting hormone, synthesized in leaves, fruit, root and seeds. Chemical nature: a 15-carbon sesquiterpenoid precursor, mevalonic acid. Physiological effect and application: induces dormancy in bud and seeds by inhibiting growth processes, accelerate the senescence of leaves, flower and fruit and their abscission, inhibits and delay cell division and cell elongation, suppresses cambium activity, causes efflux of potassium ions from the guard cell and result in stomatal closure. Thus, it functions as an anti-transpirant and act as a stress hormone. Inhibits flowering in long day plant but stimulates flowering in short day plant.